Good morning, everybody. Happy Monday. How are you doing? Well, today's Tuesday for you, but I recorded this on Monday. Today we are going to study another processes involving DNA. Last week we studied what was replication. As you could remember, replication was the duplication of the DNA. And we use it for mitosis or meiosis, no? It happened during the interphase, concretely in S phase of the interphase. Now we are going to study other two processes that are really important for our development as a living thing. We are going to study how we are going to turn the DNA, and from different processes we are going to get proteins. As I told you before, proteins are just messengers. So the DNA is the one that is going to have all the information for you, about you. For example, if you have blue eyes, or you're going to be a certain height or weight, or many things about your body, the color of your skin, many things. But the ones that are going to perform the function, to do that to happen, ¿vale? Que realmente va a realizar la función, is going to be proteins. DNA has the information. Proteins are going to be the soldiers, like the soldiers that are going to do, and they are going to perform these functions, ¿vale? Entonces el ADN lleva la información, es el que tiene las órdenes, y las proteínas son las que van a realmente a ejercer esas órdenes, van a llevar a cabo esas órdenes. Okay? So first, let's remember something from the last unit, that remember my chromosomes, my churritos. This part, each part of a chromosome is going to be a gene, ¿vale? Un gen. A gene is going to be a fragment containing DNA for a certain trait. What does this mean? That here we have a sequence, like here, no? A sequence of nucleotides. A sequence of nucleotides, and this sequence of nucleotides is going to be transformed into different proteins. Okay? So, for example, this could be the sequence of DNA for the information of the color of my eyes, brown, okay? And then there was an experiment, some experiments that these two people, Beadle and Tatum, proved, and the conclusions that they finally had were that each gene was going to codify one or more proteins, and these proteins were going to determine one trait. What we have just said, these were the two scientists that proved it. Okay, so how does it work? As I told you, we have a sequence of nucleotides. I put here the nitrogenous base, as I told you on Friday that I was going to simplify. So adenine, thymine, cytosine, guanine. Okay, here we are going to study these two processes, which are going to be transcription and translation, and due to these processes we are going to get proteins, different proteins. Okay, if something happens here, imagine that for a mutation or a mistake in these processes, we misunderstand this information and we get a different protein. Imagine transcription or translation are being done wrong. You are going to get a different protein. And if you get a different protein, there's going to be a change in the trait. This could be not a great thing, maybe, or it can be really bad. For example, there is a type of anemia, which is that genetically these processes are doing wrong. So the hemoglobin, which is the protein, right, that carries the oxygen to the cells, they cannot join to the oxygen. So these people has really big problems to have energy, and it's a very serious matter, a very serious illness. It's not the common anemia that you can have throughout your lifetime. No, it's a very specific type of anemia that is a genetic illness. And it's because the protein that has been synthesized, this hemoglobin, has now the perfect shape to join the oxygen. So imagine all the problems that these people could have. So to understand how is a protein, I have just picture here, draw here, it's like a pearl necklace. Each pearl is going to be an amino acid. We are going to have 20 amino acids, different amino acids. Arginina, trypsina, tryptophan, okay? Valina. There are a lot. Isoleucina. You don't have to learn them. So breathe, you don't have to learn them. But there are 20 amino acids. So depending on the number of amino acids that you have here in this chain, the type, if it's not the same as isoleucina, than if it is valina, for example. And the sequence, which is the order of the amino acids, remember the order of a nitrogenous base was important? It is the same in a protein. The order of the amino acids is going to give you the clue for the protein that you are going to synthesize. Okay? So basically, this is an introduction for transcription and translation that I wanted you to know. Next video is going to be about transcription, which is the first step to get a protein. What are we going to do in transcription? We are going to go from a gene, from a DNA, to a messenger RNA. Okay, but this is going to be next video. I hope that this introduction was enough to understand what we are going to study now. Okay, this is all for now. Bye bye.